and cancer. Today, Professor Cláudia will talk to us about unraveling the muscle fiber formation. And finally, we will have Professor Veronica Salerno. Professor Veronica, she studied biological sciences at Universidade Gama Filho, followed by a master and PhD in biochemical chemistry at UFRJ. Professor Veronica Salerno Pinto then did her postdoctoral training at the Macaulay Research Institute. She's associated professor at UFRJ. Her scientific interests include biochemistry of the physical exercise, non-conventional myosins and intracellular transport, physiological and biochemistry markers in response to volume, intensity, frequency of aerobic training and force. And today, Professor Veronica Salerno will talk to us about the impact of physical activity on health. So it's truly a pleasure to welcome you all, and we will start with Professor Hughes. Please, I will ask all participants except for the speakers to keep their microphones off, please, in order to make sure we all listen well. And Professor Simon, if you would like to go ahead and share your screen, that would be lovely. Thank you.
Right. Thank you very much. Can you hear me?
Yes.
Yes, all well? Good.
All good.
So thanks very much for the invitation to speak to you today. It's really great fun, and I want to talk about some thoughts on sarcopenia and muscle growth and why we would study them in zebrafish. I'm going to try and get through 3 stories, an unpublished one from a PhD student, Mike Atwaters, and postdoc Graham Attili. And then, depending on how the time goes, 2 recently published papers, one from postdoc Jeff Kellu and another from postdoc Massimo Ganassi. So we have known for 3,000 or more than 3,000 years that activity from the nerve is necessary for muscle growth and maintenance, as you can see on this Egyptian drawing. But in modern life, there's a problem for people my age, which is that our muscle tends to weaken as we get old. And one of the things that happens is we lose motor neurons, and those motor neurons then would have innervated muscle fibers. The surviving motor neurons then innervate the fibers that have been denervated, and they try and maintain them, but that system doesn't work well. Although you can do weights and you can exercise and you can maintain your muscle mass to a certain extent, the sad truth is that as you get older, your muscle gets smaller. And you can see that people differ at all ages as to how much muscle they have, but we're on a downward curve. And we know from studies from many labs and people around the world that about half of this decline is probably due to our genetics. And the other half is probably due to what might generally be classed as environmental influences. But those environmental influences can be things that happen to us at these times, or they could be things that happen to us at other times in our lives. And by and large, we know very little about either the genetics or the environmental factors that control this, except, as I mentioned, for exercise. So if we look at the legs of some people, here's a young chap, and he's got a lot of muscle in his leg. And here's an old chap who likes to drink beer and sit at the bar, and he's got not much muscle and an awful lot of fat. But as I mentioned, the good news is that if the old chap keeps exercising through his life, some of us are lucky enough to maintain our muscle. But you can take 2 people who have the same amount of muscle and do the same amount of exercise, and one will age well and another will age poorly and end up very frail. And of course, that is hugely bad for society. Well, for the individual, first of all, for their family next, because they become dependent upon much more help. Typically, old lady may fall over and break her hip, and then her muscle wastes more, and then she never walks again. And of course, for society, in Britain last year, it was about $3 billion US dollars to the costs associated with sarcopenia, aging-related muscle weakening, it's estimated. So what one would like to know is where this problem for all of us, because it does affect everybody as they get older, comes from. What causes it, what we might do about it, how we might delay it, and keep everybody more active into their older age. And a second thing that is really interesting about this difference between inactive and active people, of course, is that if you talk to sports scientists, they'll tell you that the thing that's really important to maintain muscle mass or to build muscle mass is to go to the gym and do reps. That's short rounds of high force exercise. And everybody pretty much believes that the reason why you have to do reps rather than other kinds of things like going running is that it's the high force which triggers the growth. But there's a logical problem with that view that I'll go into a little bit more in a minute. But just bear in mind that there's a general feeling that high force is driving the growth, and that means there must be some method for cells to detect that high force, and it must be working in muscle, and it must be orientated in the long axis, if you like, of the fibers to detect the forces that are being generated by the myosin. Now, essentially all cells in all animals sense force and undergo mechanotransduction, and this is a very active area of research. So, for example, the hair cells in our ears essentially detect force when we hear. Our blood vessels, if you get cardiovascular disease, you've had problems with the force, many people have had problems with the force sensing of their blood pressure and the flows inside their great vessels. And of course, we all know that if we do a bit of manual labor, the skin on our surface of our hands gets bigger, thicker. And so our cells, our stem cells in our skin are sensing force and are responding by proliferation and production of more skin. So this is a very active area of research, but we think that because muscle is a particularly active mechanical tissue, one may be able to get deep insights into mechanotransduction by working on muscle. And about 10 years ago, I began a long program of trying in the lab to try and look at this issue. So why study this issue in a zebrafish, which, you know, I would not argue that zebrafish are a good model for aging-related sarcopenia. They do get bad muscle when they get old, but that's not really what I'm going to look at. But they are an excellent model for looking at how you grow muscle, as I hope I'll convince you. So here is a 5-day-old zebrafish larva. It's hatched, it's swimming around in the bottom of a dish, and it's just about to start feeding. You can see its jaw muscles are in green, are maturing. And in red are the stem cells, the precursor cells for muscle growth and growth of other tissues in this picture. So I want to start by just doing that evolutionary bit, and in case there are any, particularly in case there are any medics in the audience, because I've discovered that medics don't get taught very much biology, and they often think that something like a zebrafish is rather similar to a C. elegans or a Drosophila. Some sort of invertebrate. Well, they're not. They're vertebrates. Muscle is a fish. And various aspects of our muscles are all shared and built in the same way. So one can use a zebrafish to look at how muscle is built and be fairly confident that you're looking at something similar to what happens in people. And the only other major model organism that's really used, of course, is the mouse, stimulated by their natural anesthetic. So, you know, this signal kind of fades away and doesn't drive the growth properly. So there's an area of time in the middle which is optimal for the growth. And of course, that's at a certain time of day. And that made Jeff Kellew, a new postdoc in the lab, a little nervous. And so he decided to look at time of day. So this is Jeff, and he produced a PNAS paper this year in 2 years of work on his own, essentially. So he's a good guy. The conclusion from his work is that circadian regulation of muscle growth drives muscle growth independent of locomotor activity. And of course, if you think about an animal during the day, it usually sleeps at night and moves around more during the day in the case of a fish or a person. In the case of a mouse, it's flipped. The mice run around at night, right? So their circadian behavior is different. And this again is a way in which fish and people are perhaps a little more similar. So let's think about circadian rhythms. This is of course, there was a Nobel Prize won the last couple of years for this. It's an endogenous biological clock that synchronizes our lives and the lives of yeast and plants and every animal with the Earth's rotation in the 24-hour period. And in muscle, there's some interesting data. So if you're an Olympic athlete, you need to get time adapted to your time zone at the Olympics, because human muscle contracts better and stronger, puts out more power in the late afternoon than it does in the morning. This is controlled, it is thought, by the clock which exists, the molecular clock which exists in all organisms, or at least all eukaryotes, I think, and is two linked feedback loops. The main one is at the top here, and is made up of two proteins, BMAL and clock, which bind to E-boxes, they're transcription factors, and they drive the expression of many, many genes inside cells, including inside muscle. And two of those genes are purr and cry. And purr and cry's function is to feedback and inhibit BMAL and clock. So you have a on and off cycle that goes every 24 hours. And there's a secondary cycle, an accessory loop, where two other targets actually affect BMAL expression. So this is complicated, and we are not working on the mechanism of the clock. But what we're interested in is, does the clock in and of itself, as opposed to the motor activity of the animal, affect the growth? So when an animal is in light and dark, genes are expressed, genes go down with a cycle. And that can be for one of two reasons. It can be a circadian clock control event, or it can be a diel cycle that's controlled by light-dark or some other Zeitgeber, as they call them, an entrainment cue. So in the case of a diel event, if you remove the light-dark or the entraining signals, then the cycling will rapidly fade out, as you see here. But in the case of a true circadian event, the circadian clock persists after you, say, leave the light on all the time, and you'll still see the oscillating behavior. And in fact, for circadian clocks, in order to distinguish them from diel light-dark cycles or other entrainment cues, there are three major criteria that must be fulfilled. The first is that they must be, I've already mentioned, the circadian rhythm persists in constant conditions when there are no Zeitgebers. The second is that the circadian rhythm should be entrainable. If you fly from London to Rio, you will get jet lag and you take a little while to adapt, but you do adapt, and then you're on Rio time. And the third one, which I'm not going to deal with, but is important for poikilothems like zebrafish, is that the clock must be temperature compensated so that you stick at 24 hours, irrespective of whether you're warm or cold. And I'll show you that the top two and basically the third one as well, but I won't show you the data for that, are all true in the zebrafish muscle case. So circadian rhythms drive cycling gene expression in muscle. This was shown first, I guess, by Karen Esser about nearly 20 years ago. We're using microarrays where about 200 or so genes changed, many of them transcription factors or signaling. Same thing done by RNA-seq now in people, in all kinds of things. Thousands of genes change. So which ones are deal and which ones are circadian? Which ones are due to the different activity that the animal has and consequent upon either deal or circadian, and which ones are really due to the circadian clock. So Jeff and I realized that we could address this in our muscle system. So first of all, for the first few slides, I'm going to show you things about deal change, just fish that are in light dark cycles. Okay, nothing about circadian yet. So we can assay the growth of fish over a day, as I showed you, and they grow by about 30%. That's the full 3 to 4 day growth. If we assay the same fish halfway through the growth period, we can see that in the daytime, the fish grow about twice as much as they do at night, in the succeeding night. So there's a deal difference in the rate of growth, high in the day and low at night. So that's kind of interesting and slightly worrying for our stimulated timing experiment, what's going on there. So muscle grows more during the day and at night, but of course the fish swim more during the day and at night. These are day 1, 2, 3, 4, 5 old fish, and somebody's recorded the activity, and you can see that they swim, they move a lot in the day and they move less when they're in the dark. But I think this exaggerates the difference, actually. There's quite a lot of movement in the dark as well. So, of course, I've shown you that we can get rid of this activity by simply adding tricaine at the beginning and keeping it there all the way through the process. And this is where Jeff's result became very interesting, because although as I've already shown you, removing the activity greatly reduced the growth, the difference between day and night persisted. So you can see that there is still significant growth during the day, where there's no growth at night when you've inhibited the activity. Very interesting. So this is really suggesting that the circadian clock, and not, or at least a deal clock, and not the activity differences, are controlling the growth. So the next thing we were worried about was, is this a developmental phenomenon? Because these are 3-day-old fish, and these are 4-day-old fish. They're 30% older, right? So they might just grow more early in their lives and slow down their growth rate later. So we can do a really cool thing with these zebrafish. We can take them and we can flip the light cycle so that they have dark in the day and light in what was the day earlier, and light later. And so simply by putting them in an incubator with the lights flipped around. And very nicely, when we do that, we see they grow more in the light phase, in the older light phase now, and less in the younger dark phase, flipping from the last time. And if we inhibit activity, we can see that that persists. They don't grow at all in the dark phase, and they do grow in the light phase. So this is either deal or circadian, but it's definitely not due to development. So then we can ask about muscle mass maintenance. You know, what's the mechanism of this process? So we all know that growth of muscle is a balance between of protein turnover between synthesis on the one hand and degradation on the other hand. And what I'll show you is that both are being regulated by the clock. So on the anabolic side, Jeff used a nice fluorescent assay for protein synthesis, which uses OPP, basically an antibiotic that blocks protein synthesis by being incorporated into nascent proteins on the ribosome and making the muscle fluorescent. And you can see that very nicely, because when we just incubate fish in OPP, their muscle goes green, and if we add cyclohexamide to block protein synthesis at the same time, they don't go green. And if we add tricaine, they go a bit green, but somewhat less green than in the controls. So activity is driving protein synthesis, but there's more protein synthesis. There's quite a lot of protein synthesis even in the absence of activity. And if we look at night, then we see that basically there's still a difference. You might not see it in the pictures, but Jeff has quantified by scanning the whole stack of the computer and then quantified the data. And you can see that here, that protein synthesis rate as assayed in this assay is lower at night. And I can't quite see what I'm pointing at here. Sorry, because of the picture of myself. Anyway, you can see on the bit I can't see that activity reduces both peaks, but there is still a difference in protein synthesis between day and night. So what this tells us is that activity is helping to drive protein synthesis, but the clock is also causing a difference with more protein synthesis during the day. Basically, I won't go through all the data in the paper. We've looked at TOR and so on. Oops, what's happened? There we are, sorry. Just lost control. Then we, what we show is that basically that extra growth that's driven in the day by the clock kind of cooperates with the extra activity during the day to drive that protein synthesis, that anabolic side. So what about the catabolic side? Because it's well known that in various animals, including people, that there's more muscle catabolism at night than in the day. And the nice thing in zebrafish is you can inhibit the proteasome by just adding this drug, MIG-132 or other drugs to the fish and then looking at what happens. And what happens is very nice. You can see that it doesn't, adding this proteasome inhibitor during the day doesn't do too much. But when we add it at night, it remarkably, and this I find very remarkable, I thought this experiment couldn't possibly work, it increases the growth of the animal specifically at night. And that is a clock-related activity, not a swimming-related activity, because it rescues specifically at night, even in trichane-treated fish. So that suggests that in the fish, in their inactive phase, there is more muscle catabolism going on, and that's part of the reason for why they grow more slowly in the night. And that's also regulated by this DL cycle, not by activity differences. So, sorry, have you lost me again?
No, we're still,
no, okay.
we're still good.
We are here.
So then the question is this the circadian clock? So the nice thing that we can do in zebrafish is we can take the embryos and we can put them into permanent darkness or permanent light from the get-go. And then they are, it was shown many years ago that they are not entrained, that they do not undergo this circadian, the clock is never entrained. And when Jeff does that, you can see that their muscles grow less well in either of the constant conditions compared with the light dark cycling conditions. So that looks like the circadian clock is kind of necessary for proper growth. But of course, the best experiment to do is to grow them in the cycling condition through the first 3 days, and then for the 4th day to put them into constant light. And now we're asking, is this a circadian phenomenon? And as you can see here, both with and without trichane, the difference between day and night persists, and therefore this growth difference is a truly circadian phenomenon. And we've shown this in the 5th day as well. So the last experiment I want to show you from the paper is to ask directly, is the molecular clock necessary for this function? And it is. So Jeff has injected into the animal RNA encoding a dominant negative form of the clock protein, one of those 2 transcription factors together with BMAL that binds to the target and drives the clock. And you can see that that stops the clock, because normally the clock cycles like this with low per1 expression. Remember, per is a part of the clock mechanism, but it's itself a target of BMAL and clock. And you can see that it normally is low at night and high during the day. But when we block the clock, it remains high all the time. It doesn't cycle anymore. And when we do that, we can see that adding delta clock reduces the growth during the day and increases the growth during the night, I think. So that delta clock inhibits anabolism in the day and it inhibits catabolism at night. So I'll skip over that, I think, and just kind of summarize by saying you can see more about this in the paper. It's a little bit complicated, but the bottom line is that during the daytime, the clock is working together with probably permissive TOR signaling to drive anabolic growth, whereas at night, the clock, there is a basal level of growth that's TOR independent that's happening anyway in these fish, and there's a clock stimulated degradative process which is kind of counteracting that and leading to less growth at night. Okay, so how are we doing? I think I've come to the end of my time, haven't I, really?
Yes.
Yes, okay.
I'm afraid so,
So
yes.
I will not tell you the last story. I will zip through this. You can read about it. It's published in eLife this year. Basically, what it showed was that we can assay in adult fish or in growing fish the behavior of the stem cells that lead to muscle growth. Because of course we think that for muscle maintenance and for muscle growth later in life, beyond a one-day time point, their action is going to be absolutely crucial. So I've shown you then in this talk that myosin function drives growth, that forcin is a downstream target of myosin which interacts with TOR to drive growth, that the circadian clock controls growth differentially in day and night, that the clock and activity cooperate to promote maximal growth, and that the clock controls growth independently of effects on activity. I won't mention those two. So I just wanted to end by coming back to sarcopenia. So when muscle grows, and we think about growth in muscle or maintenance of muscle, we have to think about three separate modes of growth. One thing which happens early in our life is the number of muscle fibers that we make when we're children. How many muscle fibers do you make? Does the number of muscle fibers you make control whether or not you get sarcopenia later in your life? Nobody knows the answer to that. My hypothesis is that if you make fewer fibers, but they may be good enough to give you quite reasonable functioning muscle in most of your life, but when you get old, they may be not that may be bad for you compared with having more fibers, but smaller. The second mode of growth is just adding nuclei from satellite cells, from precursor cells, into the pre-existing fibers. How many nuclei do you need in order to have good functioning muscle? And could we differ in the number of nuclei that we have in our fibers? And could that control whether or not, or how rapidly we get sarcopenia? And lastly, a major control on growth we know is the amount of cytoplasm, the nuclear domain size that each nucleus supports. This is rapidly changeable. Most tissues can't grow in this way by increasing or decreasing the size of their cells, but muscle can. And of course, how this mechanism operates may be very important for understanding growth. And we're looking at all of these in the zebrafish context to try and understand the fundamental basis of how you grow your muscle and what one might do to enhance it or maintain it when some of the signals are not working. So the people in yellow are the people who did the work I talked about. The work I didn't talk about was done by Massimo, which was a collaboration with Peter Zamet's lab, where he now is, my next door neighbor. This work was essentially entirely funded, I think, by the MRC, who employ me. And thank you very much for the chance to present our data.
Thank you so much, Professor Hughes. We have little time for a couple of questions. I know Manuel signed up, so Manuel, go ahead.
Okay, so first, thanks a lot, Simon. Great talk, very, very interesting. So the question is does forcing expression or activity changes during the day and night cycles?
Right, great question. We're looking at that. We don't actually know yet. We haven't seen any signal in the qPCR that we've done so far.
Okay, so just a comment. Should we sleep less to have less muscle catabolism and grow stronger?
Well, that depends. You see, what we show is it's controlled by the circadian clock. So you might want to go to Mars or something. I don't know. It may happen whether or not you're asleep. The whole point really is that activity is not what's driving this. It's the circadian clock. So no, I think the answer is probably sleep is. We know sleep is good for us, even though it wastes our lives.
Thanks.
Thank you so much. Maybe we can, if people have more questions, we will ask them in the end. Veronica, do you want to ask a question?
I'm on.
Hi.
Yeah. So I have two questions, very fast questions. First is, how do you see the question of microRNA action on muscle growth? Because there are some people that do the same exercise and the same intensity and so on, and they didn't have muscle growth, some of them, and they showed there is a specific kind of microRNA that affects us. And the second is, did you try to see when you block the muscle catabolism during the night with MG132, how is the function of the muscle in the next day? Right. Or
so about microRNA, I can say absolutely nothing. We haven't looked at microRNA, and our RNA-seq was done by throwing away the microRNAs, by size selection. So I don't have any data on that. And yes, that's interesting data. I'm by no means saying, my view is that muscle growth is is a very complicated process, and many things are going to be essential in order to drive muscle growth. And what we do as scientists is we set up the system to maximize the signal from whatever it is that we're trying to investigate, right? If we find a little effect with some treatment, we then ask, how do I do this experiment differently so I get a bigger effect, so my graph looks better, right? And everybody does that, and they all tweak their systems in slightly different ways for each thing that they're looking at, so that, one person, leucine, nutrition gives huge growth. Another person, exercise gives huge growth. Another person, the clock gives huge growth, right? The issue is, how do you integrate all of these together in one system where you can't tweak it to give a maximal effect to a particular stimulus? How does that really work in a real animal? And that's what we're trying to get at by integrating all of these things together in a manipulatable system, just in these little fish. So that's sort of where I want to go, but I've got no data about microRNAs at all. Your other question was, I've already forgotten it. What was it? Just
if you block the catabolism, the muscle catabolism.
Oh yes. Oh yeah.
Night,
No,
this doesn't mean that the muscle that was not destroyed, it's
yeah.
Good.
Yeah. No, this is why I was so surprised. I thought this experiment couldn't possibly work, right? So no, we haven't, it's quite difficult with these very small fish to really assay the function of the muscle. So what I can tell you is that cellularly, in terms of the size of the cells and the number of fibers and stuff, they look fine. When we stain for myosin and actin, they look fine. But that doesn't necessarily mean that they're totally okay. In fact, I would strongly suspect that they're not totally okay. What it does tell us is that the fibers are able to, when we block the proteasome, the fibers are actually able to go and get bigger. So they're able to get bigger, even though, so why is that? Is that because proteins that would have been required to put membrane onto the surface of the fiber are not being degraded? Proteins of that sort are in the ER, right? And so they're not really normally associated with the proteasome. There's a lot of cell biological questions that are kind of interesting to look at in this system to understand why that ridiculous experiment, putting MIG onto a fish, actually gives you more muscle growth. I just thought the fish would curl up and die when you did that, but they don't.
So thank
we
you.
We should probably move on, but thank you for your questions. That was good.
Thank you.
Thank you very much.
Wonderful indeed. Cláudia?
Yes.
Would you like to?
Yes, I will share my presentation.
Can I just ask people for the mic? Yes, it's fine. Just be aware of your microphones, everyone else, please. Thank you.
Okay, so good afternoon to everyone. Today I'm going to talk about the formation of muscle fibers. And just one second. And our lab works with two main animal muscle models. We use chick embryos and we use also zebrafish embryos. But today I'm going to talk more about the chick embryos, and more specifically, I'm going to talk about the in vitro cultures of primary myoblast cells that we do in our lab. And we have a general question in the lab. It's how muscle fibers are formed. And we have many specific questions, but today I'm going to talk about one, how different signaling pathways regulate skeletal myogenesis. And for that, I just want to talk a little bit about a general background on muscle biology. So I will be talking about a muscle cell, and a muscle cell is the same thing as a muscle fiber, and they are filled with many myofibers. And myofibers are composed of sarcomeres, and here I'm showing you one sarcomere, and sarcomeres are made of sarcomeric proteins, and there are many different proteins. The most well-known are actin and myosin, but there are many others like titin, nebulin, desmin, alpha-actinin, C-protein, myomycin, and among others. And those proteins are essential for the contraction function of muscle cells. But there is also another important steps that I'm going to talk about today. So here is one example of a muscle cell culture, a embryonic chick primary culture. And we can follow all the steps that we call myogenesis, muscle cell or muscle fiber formation. We begin usually with mononucleated cells that we call them myoblast cells. And then we have a fusion step, a myoblast fusion step, leading to the formation of early myotubes. And then these myotubes will become mature myotubes later on. And we can follow everything in these chick muscle cultures. We have isolated myoblasts, and they will elongate, they will align and fuse with each other, forming these very small myotubes in the beginning, and then they will grow, forming these thicker and longer myotubes with many sarcomeres and many nuclei within the cells. And if we look at these cells under a light microscope, these cells will contract. So they are fully capable of contraction. And in our lab, we use a lot of microscopy to analyze muscle cells. And we use mainly immunofluorescence microscopy to follow the distribution and localization of different sarcomeric and muscle-specific proteins. So like this one that we are looking at the left at an immunolabeling for alpha-actinin, staining the Z lines of sarcomeres. And we also do a lot of video microscopies to follow the dynamic aspects of cheek muscle cell cultures. So here we can see one muscle cell, one myoblast, artificially colored in pink. And we can see that the myoblast cell is rolling over a small myotube, trying to find a place to fuse. So in a few seconds, this cell will disappear and will become integrated into this myotube. So these myogenic cultures are very robust, and we have, it's easy to follow many aspects of myogenesis. And one aspect that I am very interested in is in myoblast fusion. So myoblast cells, as I told you, they will elongate and align and will fuse to form a myotube. And there is one family of proteins that have been recognized as one of the main proteins involved in myoblast fusion. And this family of proteins is the cadherin family. Cadherins are a family of proteins, and there are different cadherins involved in myoblast fusion, N cadherins, M cadherins, R cadherins, and others. And here we stained cheek muscle culture with an antibody against pancadherins. So it will label all the cadherins that have in muscle cells. And here's a specific labeling. And here there is a specific labeling for N-cadherin. We can see that the cadherins will form a line along there is someone is with the sound, sorry. N-cadherins will form these very straight lines of adhesion. So here we can see 2 myoblast cells beginning to fuse. And here we can see 3 myoblast cells, and they are engaging into myoblast fusion. And interestingly, cadherins are linked to the actin cytoskeleton, and we have one protein that can link actin to cadherins, and that's the catenin family of proteins. And in light blue, we can see beta-catenin. That's the protein that we label the cells here. And this is a very interesting protein because catenins, beta-catenin, is involved in both in cell adhesion during myoblast fusion, but it's also involved in the Wnt beta-catenin signaling. That's the one I'm talking about today with you. So today I selected 3 different stories to tell you. Simon did talk about 2 different stories. I will try to talk very quickly about those 3 because they have something in common, and I will talk to you at the end. They have the Wnt beta-catenin signaling linking all of those studies. So my lab did some studies in the last years, studying membrane microdomains, lipid rafts, and membrane cholesterol. And then we did also some studies using the LMO7 protein. And more recently, we began studying lysosomes and LAMP2 protein. LAMP2 is a lysosomal protein. So I will begin by showing you just the first study on membrane cholesterol and lipid rafts. So my lab have been working with lipid rafts for more than 10 years. And we did a lot of work with these students, Débora Portillo, Carolina Pontes, and Eliane Ribeiro. And we found that when we change the membrane composition, when we deplete cholesterol, we can enhance the formation of myofibers. The fibers will be larger than usual, and the whole process involves the Wnt beta-catenin pathway. So just to tell to this broader audience that we have here, a signaling pathway is a transduction signaling mechanism that we have in our cells. For that, we need an extracellular signal, a transmembrane receptor, an intracellular signaling proteins, and then one protein could be translocated to the nuclei and will regulate gene expression. And then the cell will respond either by proliferating, survival, or differentiation. Today, I'm going to talk about muscle differentiation, and the cell signaling pathway that will be focusing will be the Wnt beta-catenin pathway. So the Wnt pathway is one signaling pathway among many others that we have in our cells. And just to show you the variety of signaling pathways, and they also interact with each other, which makes things more complicated. And we have in muscle development, we have 3 major signaling pathways that can have a major role during the initial steps of myogenesis. And these signaling pathways are the Wnt pathway, sonic hedgehog pathway, and BMP pathway. So in this cartoon, I'm showing you receptors for each of these signaling pathways. I'm showing you also the extracellular signaling molecules for each of the pathways, and they all converge into the activation of one gene that we call MyoD. MyoD is a master switch gene that's involved in muscle commitment and muscle differentiation. And MyoD can in turn activate the expression of many different muscle-specific proteins that are, some of them are myofibrillar sarcomeric proteins. So they all lead to this MyoD concentration of responses. So the Wnt signaling pathway, we have this pathway in a turning in a on situation, or the pathway can be turned off. In order to have the pathway on, we need the extracellular signal molecule, the Wnt molecules. And Wnt molecules are, again, a family of proteins. We have a number of different Wnt molecules in eukaryotic cells. And we have also 2 different receptors for this pathway, frisel receptors and the co-receptor LRP56. And we have, again, a number of different frisel molecules in muscle cells and also in non-muscle cells. And this signaling, in order to be turned on, we need Wnt molecule to bind to the receptor and co-receptor, and this will activate a signaling, intracellular signaling cascade involving these proteins, JSK3 beta, axin, Dishevel, and APC, among others. And then this will lead to the final event, that's the translocation of beta-catenin to the nucleus of the cells. And beta-catenin will then regulate gene expression, activating many genes, including MyoD, and then including many myofibrillar proteins. So beta-catenin is highly involved in muscle development. But things are never simple in biology. So besides the canonical Wnt pathway, there are others non-canonical Wnt pathways. The interesting thing is that beta-catenin, it's dependent, it's part of the canonical pathway. This pathway depends on beta-catenin, and the other non-canonical pathways are beta-catenin independent. So today I'm going to concentrate on one Wnt molecule that's part of the canonical Wnt pathway, Wnt3A, and one Wnt molecule that's a representative of the non-canonical Wnt pathway, that's Wnt5A. So what we did in our lab a few years ago is that we decided to test, to probe different molecules or drugs that could activate or inhibit the Wnt beta-catenin pathway using these chick primary muscle cultures. So we decided to use 2 activators and 2 inhibitors of the Wnt beta-catenin pathway. The activators were Wnt3A, that can bind to the receptors and activate all the signaling cascade. And we also used BIU. BIU is an inhibitor of the enzyme JSK3 beta. And when the enzyme is inhibited, beta-catenin is now free to be translocated to the nucleus. So we used these 3 activators and 2 inhibitors. One was the DKK1, which inhibits the co-receptor LRP56, and in this way prevents Wnt binding to the receptor. And the other inhibitor is called Frisbee. Frisel is like a similar molecule as compared to the frisel receptor. And frisel will then bind to Wnt molecules and prevent Wnt binding to the real receptor at the plasma membrane. So those 4 molecules were tested in our chick myogenic cultures, and I will show you in the following slides. So Wnt3A, we found that it promotes the enlargement, the growth of muscle fibers. We could see in the culture very large and thick myotubes, whereas Wnt5A, the non-canonical Wnt molecule, inhibits muscle formation. So we hardly could find myotubes in the same size as the control culture. We also showed the same effect of Wnt3A using immunofluorescence microscopy, just to show that the myotubes were completely filled with sarcomeric structures. So was a fully contracting and normal myotube, but a very thick one. So we also tested the inhibitor, Frisbee, and we found that Frisbee could inhibit muscle formation, muscle fiber formation. And we also tested the other inhibitor, DKK1, and we found again that DKK1 completely inhibited myofiber formation, as you can see here. And finally, we tested the effects of BIO, which is an activator of the Wnt pathway. And here I'm showing you a different analysis. We just analyzed the number of nuclei in the whole culture and the number of cells within myotubes. And we saw an increase in myoblast proliferation and also an increase in myoblast fusion. So here we have the whole results. They are all shown in the same graph. And you can see that we have myoblast fusion in the control situation. And when we treat cells with either BIO or Wnt3A, both are activators of the Wnt beta-catenin pathway, the canonical pathway. We have an increase in myoblast fusion and an increase in myotube size, myotube thickness. And when we treat cells with Wnt5A, which is in the non-canonical Wnt representative, and also the inhibitors of the canonical pathway, they all inhibited myoblast formation, fusion, and also the formation of myotubes. Those analyses were made by the percentage of nuclei contained in alpha-actinin positive myotubes, or also with myosin heavy chain positive myotubes. So after this analysis, we decided to do an RNA-seq, a transcriptome analysis of chick myogenic cultures. And we found that the most frequent mRNAs found in these cultures was of the protein LMO7. I will tell you a little bit about the protein in the next slides. But also we found a number of mRNAs related to proteins present in lysosomes. And then those will be the two next stories that I'm going to tell you, LMO7 and lysosomes, and that's why those stories are all connected. So to tell you a little bit about this LMO7 protein, this is a protein that's very interesting, very similar to beta-catenin in the way that LMO7 can be found at the cell-cell adhesion sites. So in the sites where a myoblast will adhere to another myoblast cell, but LMO7 can also be translocated to the nucleus of cells and also can regulate gene expression in a similar way as beta-catenin do. So it has been shown that in C2C12 cells, the cell lineage C2C12, LMO7 is particularly found within the nuclei of cells. It's regulating gene expression at the myoblast phase. And when myotubes or muscle fibers are formed, LMO7 exits the nuclei of these cells, and it's found in the cytoplasm of the cells and also in the perinuclear area. So there is a shift from LMO7 localization from the nucleus to the cytoplasm along myogenic differentiation. So we decided to analyze if this is the same situation in chick myogenic cells. So we do some immunolabeling of LMO7 in these cultures, and we found exactly the same situation as was described in C2C12 cells. LMO7 was found concentrated within the nuclei of mononucleated myoblast cells and was found in the cytoplasm and and in the perinuclear area of myotubes or muscle fibers. And here I show you in a cartoon, LMO7 concentrated within the nuclei of myoblast cells and in the perinuclear area of myotubes. We decided to downregulate LMO7 in chick muscle cells, and we designed 3 different siRNAs to inhibit the translation of LMO7. And when we did that, we found that LMO7, when we have a downregulation of LMO7, we also found an inhibition in muscle fiber formation. Cells that lack LMO7 translation showed very thin myotubes. We saw less myotubes, and the few myotubes that we saw, they were thinner than the control situation. So LMO7 could inhibit myogenesis. So we decided to test whether we could rescue the LMO7 phenotype using either Wnt3a enriched medium or bio. Just remember that both are activators of the canonical Wnt pathway. So what we did was that we knocked down LMO7, and we did also another experiment that when we transfected cells with the sRNA for LMO7, we treated cells at the same time with Wnt3a or with bio. And we found that both Wnt activators could rescue the muscle formation phenotype. So Wnt beta-catenin pathway could rescue the absence of LMO7 in these chick myogenic cells. So those results were done by my former student of the lab, a PhD student, Ana Cláudia Possidonio. And that leads us to the third story that's related to lysosomes and LAMP2. Remember that I told you that lysosomes are among the most expressed mRNAs in chick muscle cells during the initial steps of myogenesis. Just a little background about lysosomes. They are not only organelles involved in macromolecular digestion, autophagy, endocytosis, phagocytosis, but they are also cell signaling platforms. This is a 10-year story. There are many papers today in the literature describing lysosomes as hubs for cell signaling. So many different signaling pathways use lysosomes to degrade some of the intracellular machinery of some of the signaling pathways, including mTOR, Wnt signaling pathway, among others. So we decided to do 2 strategies. The first one is that we decided to downregulate the expression of LAMP2 in these chick myogenic cultures. Why we chose LAMP2? Because LAMP2 is one of the major proteins present in lysosomes. It's a transmembrane protein present at the membrane of lysosomes and is also involved in ionic transport. So we downregulated LAMP2 in these cultures using the same approach, siRNA to LAMP2 protein. And what we found was a similar result as compared to LMO7. When we downregulated the expression of LAMP2, we found an inhibition of myogenesis. We found a less number of myotubes, and the myotubes were thinner than the control situation. But we also decided to do inhibition of lysosomal function. And for that, we used a lysosomal inhibitor called Lys5. Lys5 is a chlorokine and inhibits all acidic compartments in cells. And what Lys5 did was Lys5 inhibited the formation of muscle fibers and also inhibited myoblast fusion. And again, interestingly, we decided to try to revert this phenotype by treating cells with Wnt3a together with Lys5. And again, we found the same phenotype as we found trying to rescue LMO7 deficient cells. We could revert the Lys5 effects on myogenesis by using an activator of the Wnt beta-catenin pathway, specifically the canonical pathway. So we found that very intriguing. Those results were published this year by 2 students of the lab, Caio Bagri, which is a PhD student in the lab, and Ivone Rosa, which is a PhD fellow in the lab. And these 3 stories have, what they have in common is that all of them we found that Wnt beta-catenin pathway could rescue those phenotypes. So we now have 2 hypotheses to try to understand what's going on, why in cheek muscle cells we can revert or recuperate all the phenotypes using the Wnt beta-catenin pathway, only the canonical and not the non-canonical. So is that a very strong, in a way, a very robust signaling pathway? Maybe in muscle cells, the Wnt beta-catenin pathway is the major pathway, is somehow more, I don't know, more relevant than the others. Or we have also other options. So we have now 2 mechanisms that we are beginning to analyze them deeper. So I don't have specific answers for that. And I decided to bring these questions here today because this is something new that we are now working on in our lab. So the first mechanism is that maybe membrane cholesterol, lipid rafts, LMO7, lysosomes, and LAMPs, they are interfering with different subpopulation of these myogenic cells. I didn't tell you in the beginning, but cheek myogenic cells have different phenotypes in these cultures. We have fibroblasts coming from connective tissue. We have replicating myoblasts. We have postmitotic myoblasts. We have some bipolar myoblasts that are the ones that are elongating to engage into myoblast fusion. And we have myotubes or muscle fibers that are already formed. And we have also some quiescent cells, cells that resemble satellite cells. People call them quiescent cells or reserve cells. So maybe the Wnt beta-catenin pathway is rescuing all these phenotypes, but maybe acting in different subpopulations. We don't know that yet. And another possible explanation is that maybe those molecules and those different drugs that I'm using are interfering with the Wnt beta-catenin pathway at different points of the signaling. Remember that we have Wnt3A binding to these 2 receptors and then engaging these intracellular molecules. And this will all lead to beta-catenin inside the nucleus. And then beta-catenin itself will activate MyoD and left TCF, and they will both engage into the activation of those myofibrillar and muscle-specific proteins synthesis in the cells. So why this only one molecule like Wnt3A can have a very strong phenotype in this muscle cell, chicken muscle cultures? That's why we have these 2 mechanisms. I don't know which one is the correct, and I will be glad to have some questions or insights from you to help us in this new phase. So we thank all of our lab group, former students and present students, and thank you very much for your patience. Thank you, Catarina.
Thank you so much, Claudia. Maybe we have time for one little question.
Okay.
Then we ask in the end. So
I have a question. May I?
Sure.
Please.
Okay, so nice talk, Cláudia. My name is Desvendando. I'm from the Federal University of Minas Gerais.
Mm-hmm.
And I have 2 questions about your talk. That's very interesting. I have many questions, but just to don't make any problem in your schedule. But
Okay.
how LMO7 is acting to control the phenotype that you said here? You imagine how, I mean, the protein that's regulated by this transcription factor, because this protein acts as a transcription factor, no?
Yes, yes.
So you imagine the target?
Yes, very good question. I had a little time to present everything, so I passed it very quickly in many of the results. So LMO7 is described as having some similar targets within the nuclei as compared to beta-catenin. So there is one paper saying that LMO7 can compete with beta-catenin within the nuclei to bind to both MyoD and left TCF. So there are more than one paper showing that LMO7 acts like regulator of gene transcription and using the similar sites as beta-catenin. I'm not sure if this is the same thing that's happening in these cheek muscle cells.
But you don't have measured these myogenic factors.
No. In this work, no. We just saw that we have more, when we treat cells with Wnt3A, we have an increased expression of beta-catenin within the nuclei of these cells. But we don't know if, and probably we didn't test, but probably also MyoD and all the proteins, because we have a fully formed myotube filled with actin, myosin, titin, and so on. But we did not do a PCR to test.
Okay. And you showed some results about the inhibition of lysosome to study myoblast fusion and growth. But how can you imagine that you can dissociate it between the effects on protein homeostasis, the proteastasis, and the inhibition or stimulation of some intracellular signaling pathways? Because we know that when we hit out lysosome and autophagic lysosome pathway, accumulates some aggregates protein inside the cell, and this toxic to the cells. So what do you think about it?
Okay.
Or even remove some protein that's important in the process?
Yeah,
what do you say about that?
Yeah, very good question. So Lys5, in the beginning of the work, we tested different concentrations of Lys5, and I didn't show here the viability tests. And Lys5 can kill all the cells when we do, if I'm not wrong, 10 micromolar concentration. So we decided to use a concentration where we could affect cell viability in a very low level, like 30 to 40% at the maximum. So we don't kill off all the cells, because we still have lysosomes, but we are also only down-regulating the appearance of lysosomes. So we have less lysosomes than a normal cell, but we still need to have some, because a cell needs to still, as I told you, needs to do the normal micromolecular digestion, autophagy, and so on. So this is a very fine regulation. The concentration of Lys5 was really critical in the beginning of the work. Okay, and we did also many other control experiments analyzing all the acidic compartments of the cell using acridine orange to see how the other compartments are still functioning. But you are right. Thanks. Yeah, we still always have the problem of we need lysosomes and we can't get rid of all of them. Okay.
Okay. Thank you, Claudia.
Thank you.
Thank you very much.
Thank you for your question. Thank you, Claudia, for your nice talk. I would like to invite Professor Veronica Salerno to please share her presentation with us?
It's working?
Yes.
So, thank you very much for the invitation to participate of this talk and this conference. It's a pleasure to be here. And today I will talk, I will give you an overview about the impact of the health impact of exercise on health. So to start this, I would like to show you this. Whoa, not going. Okay. This graph that shows all causes of mortality reduction versus daily physical activity duration. If we look at this graph, we can see, for example, that 60 minutes per day of a low intensity exercise like walking is capable to reduce around 20–25% of all causes of mortality. If we start doing some more vigorous exercise and reduce the time, for example, 30 minutes, we saw a reduction around 30–35% of all causes of mortality. With this graph, we can have a good idea how important is the exercise to keep health. And I can tell you, exercise is a polypill. It's a health polypill. So why exercise is able to do that? Exercise is a kind of challenger for our system, and it affects all our systems, not just one. If we are doing some aerobic exercise, sorry, we have effect on central nervous system, in metabolism, in oxygen transporter, in skeletal muscle. We challenge all these systems, including the skin. And it changes the metabolism of these systems to keep the exercise. And this kind of stimulus or good stress, when the exercise is done in a right amount of exercise, intensity and duration, the exercise causes different stimulus like mechanical load, increase oxidative stress, reduce tissue oxygenation, ATP turnover, calcium flux. And these stimulus, this kind of stimulus will activate some important pathways, signaling pathways. And these pathways will answer increasing mitochondrial biogenesis, protein synthesis and degradation, fiber formation, hypertrophy, as we saw, autophagy, increase the antioxidant defense, and cytoprotection. So it turns our cells, our systems, more healthy. Exercise, for example, here we have two different kinds of exercise, anaerobic exercise and the strength training exercise. The main effect, but not only, the main effect of aerobic exercise is activation some signaling pathway that increase mitochondrial biogenesis, for example, increase glycogen and triglycerol in the fibers, muscle buffering, and capillary density, what is very nice because it increases the support or apport of oxygen and nutrients for these muscles. But not just only this, this kind of exercise increases the synthesis and the translocation of GLUT4, for example, a transporter of glucose, to the cell membrane. And it's very important, mainly for people that have diabetes or type 2 or type 1 diabetes, because the exercise will help these persons to keep the glucose level in a good range. Also, it increases the CD36 fat acid transport, what improves the use of fat acid during the metabolism, reducing, for example, the storage of fat acid in white adipose tissue, avoiding, for example, helping to avoid some metabolic disease. By the other side, we have another kind of exercise that is strength training, that the main signaling pathway, but not only, the main signaling pathway of this exercise is improve, for example, protein synthesis, what will cause muscle hypertrophy. And it's very important to health, because if we have more muscle, we increase the metabolism, the basal metabolism, avoiding, for example, the deposit of lipids on fat tissue in adipocytes, for example. So in this way, we can tell that exercise in terms of systemic effects, it affects skeletal muscle causing hypertrophy, affect the vascular, improving flow, vasoreactivity, and angiogenesis. What's very important also, because it helps to control and avoid hypertension, for example. It improves the metabolism, what's good for diabetic persons or people that to avoid overweight. It's for, in terms of cardiac effects, we also have a huge effect of exercise in cardiac muscle, increase cardiac growth, cause cardioprotection, improve the function like increasing stroke volume, cardiac output, increasing the cardiac metabolism and vascular in the cardiac muscle. So for health people, it's preventing disease. Exercise is preventing disease, turning our systems in a better working form. But also exercise can be used to help people that has some, for example, cardiac disease. Here we have a dilated cardiomyopathy, ischemic heart, or aged heart. If people doing exercise, aerobic exercise, that's the most recommended to this kind of diseases, we will observe an improvement in the heart function, increasing autonomic heart rate control, stroke volume, cardiac output, also increase mitochondrial function, reduces cardiac fibrosis, reverse the left ventricle remodeling, and it helps with arrhythmias. So exercise helps prevent and helps in the treatment of heart disease. But if we look, the majority of the heart disease death, we can see that is associated to ischemic heart problems. Here we have 46% in men and women, 38% of ischemic cardiac disease death. So what is this? This ischemic cardiac disease is caused by a block in some artery that irrigates some part of the heart. With this block, we have the increase of reactive oxygen species, ROS, calcium, calpain, protons, and of course, we reduce the ATP synthesis, causing cell death. But the worst part, what is more dangerous or cause more cell death, is when we have the reperfusion. Because when we have the reperfusion, we have a burst of oxygen, and it increases a lot the amount of reactive oxygen species, calcium, calpain, caspase. And it will culminate with mitochondrial lesion and cell death. So in our lab, we are trying to, one of the things that we do, we are trying to investigate if the exercise can protect against this process. So we use exercise, and look, here we are using not a training, we use just 7 days of low intensity exercise. And as we can see here we measure the infarction size. The exercise after 7 days protects the lesion of the heart. And not just this, it improves the left ventricular pressure developed by the heart. It protects the cell and gets a better function during the hyperfusion compared with the control here. But also, we try another strategy together with alone or together with the exercise. We use natural beverage that's very common in South America, is a tea called erva mate, that has a huge amount of antioxidants. Why this? Because as I show you, ROS is one of the worst things in this scenario of ischemia reperfusion. When we use just tea, the animals had a reduction on the infarction size area and a better function of the heart during the reperfusion. But when we mix these two strategies, we lost the effect. And the question was, why? So there is a kind of stress called reductive stress. It means when we use a high amount of antioxidants, we block the adaptation of the tissue, and also we cause oxidative stress in this tissue. So when we look this, we saw that in the presence of the tea, we have a lipid peroxidation was increased, as well as the protein carbonylation, showing that in the presence of high antioxidants, we have relatively bigger amount of oxidative stress that cause a precondition in the state and protects the heart. And it's not seen in the other two conditions. So when we evaluate this to see what exercise was doing, we saw that exercise does the opposite thing that antioxidants. It increases the activity of antioxidant enzymes protecting the heart. And when we have a huge amount of antioxidants, we don't see this adaptation and the effect is lost. So here I'll show to you that exercise prevent, protect, and can be very helpful during a cardiac event because it protects against the lesion, reducing the lesion. And look, it was just 7 days of exercise and low intensity exercise. Another point that I would like to talk, it's about the aging. Aging is a condition that increases the health problems. Why? Because we have, with aging, we have deleterious effects in many systems. We reduce brain function, cardiac function, lung, muscle, we change body composition, and also we reduce the metabolism. And exercise is very important for this process to avoid this deleterious picture. It increases, it reverses this. For example, we achieve our they peak our physiological function around 30 years old. After that, we lost around 1% per year. And if a person is not sedentary, if a person is an active person, it delayed this process for 20 years, and it will start to lose around 50, the physiological function. And better, we will be in a high condition in terms of physiology. We will start to lose in the up state, not in a down state as we saw in sedentary people. Another point here, so I can tell also that exercise is a youth pill. But another problem nowadays is not just people are sedentary, it's the caloric intake from everybody. We ate much more than we need. And of course, exercise try to counteract this, but we need to give a little help. There is I will show you some data in this aspect. And there is a famous article showing that these two monkeys and this C and D, they had a reduction of 20% of caloric intake. They have the same diet, but just 20% of caloric intake. And as we can see here, this one, they had a caloric restriction, look much younger, although they have the same age. And also, caloric restriction reduce cancer, neurological disease, metabolic syndrome, and reduce the production of reactive oxygen species that cause the cell death. And it's increased during the process of aging. So, in terms of metabolism, it's very good. So we think if caloric restriction improves the metabolism, as well as the exercise improving the metabolism, if we combine both strategies, we will have a better function, a better metabolism for the animals. So during the aging process, for example, so we did this, and here we can see that the weight of these animals under caloric restriction they didn't gain weight, as well as the group that combined caloric restriction and training. Here, the training was different from what I showed before. It was a high intensity interval training. It's shorter and high intensity, and in a high intensity, and have the same, the literature show it has the same effects of moderate intensity exercise. So here we can see the difference in terms of weight. And most of this is associated with the loss, the reduction of fat mass. When we look at the skeletal muscle, we can see that the combination between high intensity interval training and diet, caloric restriction, improved the cross-sectional area of the muscle. So the muscles are better. It means that probably these animals can have a better performance. When we measure it, we saw that animals that are under caloric restriction and training, they had a better performance than the animals that are just training, showing that the interaction between these two strategies can help a lot the health during the process of aging. And why it's occurring? So probably it's turning the metabolism better. So the first thing that we did was measure the glycemic, the glucose level after 12-hour fasting. And we didn't see any difference. But when we did glucose tolerance tests, we saw that in the presence of caloric restriction or the combination of caloric restriction and exercise, we had a better use of glycose, as well as a reduction in insulin secretion. What's very nice. Imagine a person that is diabetic, that has diabetes. It will be very good for them. So, but if it's changing this, maybe it could be changing also or improving also the metabolism. So to answer this, we measure the activity of the first enzyme of the glycolytic pathway, exokinase, in 3 different tissues: liver, heart, and skeletal muscle. As we can see here, in liver, the exercise improved the activity of this enzyme. The diet also improved the activity. And when we combine the exercise and the diet, this gets even better. In heart, the main effect over this enzyme is caused by diet. But when we combine the diet with the exercise, it gets better. In skeletal muscle, the exercise improves the exokinase activity. But when we mix these 2 strategies, it gets better. So it's using the glycose better, in a better way, apparently. We measure also, I didn't bring this data, but we measure also the PFK activity and concentration, and we saw that in this case, it was higher. So the question is, it's just turning the glycolytic pathway better, or it improved all range of production, ATP production? To answer this, we measure the mitochondria respiratory pathway. So here I bring to show you the oxygen flux during the stage 3 of mitochondria respiration, that is couple state, a productive and couple state. So as we can see here, exercise improve the oxygen flux in the mitochondria, as well as the diet. But when I mix each other, it gets better. So if we reduce here, the reduction of caloric intake was around 15%. We have a better improvement of respiratory pathways. And also the control, respiratory control of the mitochondria get better. If we look the diet and the diet combined with the exercise, it gets much better. It shows us that the mitochondria is more coupled, it's more efficient. But mitochondria is more coupled and more efficient, but the enzyme that does the ATP synthesis is answering the same way. So to answer this, we measure the F1, FO ATPase activity. And as we can see here, in the presence of these 2 strategies, caloric restriction plus high intensity exercise, the ATPase activity of this enzyme is higher. It shows us that we have better, or we are improving the ATP synthesis, not just the flux of electrons by the complex, but also the synthesis of ATP. And as I showed you, mitochondria is more coupled. If mitochondria is more coupled, I expect has a less production of superoxide, that's one oxygen reactive species, and then I expect have a less oxidative stress. to confirm this, to evaluate this, we measure some biomarkers of oxidative stress. Here's a lipid peroxidation, and we can see that in the presence of diet and exercise, we have an important reduction of lipid peroxidation. And also, protein carbonylation was reduced by exercise and by the combination of exercise and diet. In this way, I can tell you that exercise improves the metabolism. And if we associate the exercise with a reduction of caloric intake, we will still have a better metabolism, what improves a lot the health of elderly people. So, thank you very much. This is my lab group, and this is Clarice and Katia, and also Antonio that work with oxidative stress. And here is Camille and Fabio that work with ischemia and hyperfusion exercise, the effect of exercise on the heart. Thank you very much.
Thank you very much, Professor Veronica Salerno. We are now open to questions. Actually, not just for Veronica, first for Veronica, but maybe if people have questions for the previous speakers as well. So please.
May I start again?
Please go ahead.
Okay. Nice talk, Veronica. Thanks a lot. And I have a question about the fiber size that you showed in the animals that were submitted to caloric restriction and exercise. If I'm right, you showed that caloric restriction exacerbate the effect of exercise on fiber size. I'm right? You show that?
No.
Or I mean,
not exactly this, because
in this one.
See, here I have an improvement of cross-sectional area.
Yes.
I believe in terms of these results, as well as, for example, for mitochondria, that under caloric restriction, you need to have a more efficient pathways and metabolism and organelles. So I don't know exactly here because I do not work exactly with muscle hypertrophy, but I believe that the combination of these two strategies triggers some better control of muscle. I don't know if it's higher because we had more synthesis, or we reduced the catabolism. I don't know.
Yeah, but this I think that's an unexpected result, no? Because it's a little bit strange that you have, because this is something that for me is very clear. Exercise and fasting are pretty similar in the sense of metabolic changes and also hormonal changes. So you have many changes in the organism that's very similar between exercise and fasting. So here you don't have a fasting, it's a caloric restriction, but anyway, you are inducing many changes in the body, like insulin that's reducing. I imagine that's you showed a result about insulin levels, but it's not lower in the group of caloric restriction, or isn't it? I don't remember. Just in this group. Yes, and this aspect as well. So if you have less insulin, and I suppose that you have maybe more corticosterone in the blood of these animals, so you have catabolic signals, not anabolic signals.
I don't know.
So it's, yes, it's,
yeah,
but it's something that's interesting. Anyway, it's something that's very interesting. So it's just something that I was thinking here about to understand the molecular mechanism that's involved in this effect.
But as I told you, I think, and people that have been working in this area, they think that although the caloric restriction reduces the nutrients, but do not cause bad nutrition, right? It turns the body in a more efficient way. You have a more controlled levels of hormones, enzymes, and so on. When I started this project many, many years ago, I expect to have a loss of performance, a loss of muscle mass, but it's not true. And when I start to look more careful in the literature, there are some athletes that use caloric restriction to get a better performance, you know? So it's very intriguing, and we start to try understand why, or which is the signaling or the main thing that turns this result so different from what everybody expects.
Yeah, and I think that's most interesting, because in the endurance performance is expected to improve because the effect on mitochondria, but muscle growth with caloric restriction, for me, this is the most surprising data.
Yeah, and we have some weightliftings, the guys that work with strength training that use caloric restriction and also avoid meat, for example.
Okay.
They are vegetarian, and they have a very intriguing and good results.
Yeah, yeah. Okay, thank you, Veronica. Thanks.
You're welcome.
Thanks.
Does anyone else have a question for Professor Veronica?
Maybe I could just follow up on that. So
please.
If I understand rightly on caloric restriction, essentially the animal is given a standard amount of food. So, and will eat all the food that it's given, I assume. Is this correct?
No. In this kind of caloric restriction that we did was every other day diet. So they have food for 24 hours and they stay without food for 24 hours. Because we choose this because it's a little bit more easy when we work with animals. And we control how much they had the reduction of caloric intake. And in this case was around 15%. And just this.
So maybe my question isn't really worthwhile, but what I was, so did you see in the exercised versus the unexercised calorically restricted animals that they ate the same amount of food in total?
Exercise and the control.
No, exercise and caloric restriction alone versus caloric restriction plus exercise.
Yes.
So, okay, so then if they're consuming the same total amount of food and they're doing a lot more exercise, doesn't this mean that their basal metabolic rate for everything else is going down? Did you measure their basal metabolic rate?
No, we didn't measure it. We are performing those experiments and metabolic cage.
Right. Because obviously the energy has to come from somewhere.
In two different kinds of caloric restriction. I mean, eat the other day and also reducing the amount of food, because this kind of diet, eat the other day, the animals, the day that they eat, they have a hyperphagic behavior during around 1 hour. And also they have some changes in the hormones that control the appetite. So what
would be very interesting
happen with other kind of diet? So we are interested to compare both and see what happened, and in terms of metabolic basal state or using nutrients.
Because if you did a restricted diet where they ate every day but a small amount, then that might answer David's question differently. Yeah.
Yes.
Very interesting. Very interesting. Thank you.
You're welcome.
Manuel?
Veronica, nice talk. I was just trying to mix everything we heard about, and I was wondering, since you have the circadian rhythm is something intrinsic to the genes, would the cells in culture preserve the rhythm? Would they have changed day or periodical changes in gene expression, for instance? Could Cláudia be able to see, if she look at different time points, could she be able to see different levels of activities in culture? And I was wondering, because and if you could do in culture caloric restriction, or give less media to the cells and see how if the cells behave differently in culture.
Yeah, it's possible. I think there are some papers showing that reducing the amount of serum, for example, in the plaque, you have similar effects to what I'm showing they didn't measure exactly this all, but they have similar effects, and there are some experiments going on for using the serum of these animals in a culture and see if there are something there. Because another thing that I didn't talk because it's too fast, it's 20 minutes, it's too fast, and English is not my first language, as you all can see that. The muscle, when we do exercise, muscle release some myosins that are hormones, has hormones, functions, and maybe we can have some different liberation or releasing of these myosins in association with exercise and caloric restriction. We don't know yet. It's relatively new. Although caloric restriction was described for the first time in 1945, the first paper, or 35, I think was 45. It didn't move a lot. Just the last 15, 20 years, it started to get more strong, the results and the research in area.
Okay, I guess we have...
If I can say something about the circadian aspect, Manel, I was wondering about that with Cláudia's experiments as well, but the story with the clock is a little bit complicated. So when you don't have entrainment cues, so long as the molecular mechanism of the clock is okay, it is thought that the clock operates cell autonomously in every cell and goes out of phase between the cells. So, so you might expect that would be what would happen in the incubator. But on the other hand, if you open the door of the incubator and you cause a temperature change, that is known to be an entrainment cue. So anything that sort of synchronizes things for the cells can actually bring the clock back into synchrony. And so, of course, because people tend to open the door of the incubator during the day and not in the middle of the night, you have a kind of way of the cells becoming entrained. So we had to get a special incubator for Jeff and actually measure the temperature change, and only changes 0.1 degree over the whole 24 hours, which we were happy about. But they say about 2 degrees change is enough to entrain.
Very interesting.
We have some questions showing up in the chat. So, David, if you want to go ahead, and then Claudia.
Yeah,
okay. Please.
Yeah, just one question to Dr. Simon. Again, thanks for your talk, brilliant talk. Very interesting, your results. I'd like to know about mTOR that you said in the end of your presentation that maybe there is some interaction between forcing and mTOR, but anyway, when you inhibited mTOR with rapamycin or taurine that you used, if I remember very well, I don't remember. Well, I think that was taurine, no? Yeah. So the mTOR inhibition blocked the muscle growth induced by the electrostimulation, no? But yes, you couldn't see these results.
A reducing effect on the stimulated growth, but not on, weirdly, not on the growth, or not very much effect on the normal growth of the animals when they're just normally swimming. So we don't really understand why that is.
Okay. So I think that I missed this point in your presentation. So
yeah, I mean, I showed it in the other order. I showed that putting in taurine didn't affect normal growth, and then I showed the stimulated growth and where there's this reduction, which we were, it's not clear that it's as big a reduction as the myosin inhibitors do. It might be a lesser reduction, but my idea, our working model, is that either TOR can also affect forcing. That's a possibility. But the activity isn't working through changing TOR, we don't think, in this situation. The muscle contraction isn't working through TOR. So either that's going on, or what TOR is doing is one of two other things. It might just be permissive. When you put in an inhibitor and you completely block the pathway, maybe that just messes up cells in quite a big way. And, you just need some level of TOR activity for the cell to be okay enough to respond to forcing. Or it could be that forcing is some kind of gain control. Sorry, TOR is some kind of gain control on the forcing pathway. So that if you have no TOR at all, you've got no gain, you're multiplying by 0 and nothing happens. But if you turn it down a bit, then you're multiplying by a lower number, but you still need the forcing signal. So we're trying to sort those things out.
Okay, and about AKT, that's upstream to mTOR, that we know that's another kinase that's very important to muscle growth, that can induce muscle growth independently of mTOR. So what do you say about that?
Yeah, so we looked at that, and there's no change in AKT activity that we can see at all under any circumstance. Yeah, we don't think that AKT is involved, and therefore we don't think that IGF and other upstream things there are likely to be involved. But of course, until you do real loss of function genetics, you can't formally prove this. So I don't want to say there's nothing happening, but we've looked and we can't see much suggestive of, by looking at phospho-AKT and so and things aren't changing.
Okay, thank you. Thank you.
Claudia?
Yes.
You have a question?
Yes, Simon. I was thinking that zebrafish, when we see some works that people do in the wild in India rivers, I saw something that the environment can change a lot and the rivers can be very dark. And so there is no light going when the fish is swimming. So do you think that during evolution, how the circadian clock could work? Will be like a second plan B?
Right.
Yeah.
So it's good question. I mean, I strongly suspect that the fish swim up to the surface sometimes, and there will be other things. I don't know if you know, but in the circadian field, there are all sorts of exciting stories. So one of them is the story about the bat poo. So there are these blind cave fish that live in, they're blind, so they don't see any light, and they live in these caves in Mexico. And they still have an entrained circadian clock, which is entrained to the sun, even though they live permanently in total darkness. And it turned out when people analyzed this, that it's because every night the bats come into the, or every day, I should say, the bats come into the cave and they poo in the water that the fish live in, and that feeding stimulus is enough to entrain the clock for the fish. Okay. So I don't think there's a problem with getting entrained in a paddy field with a lot of muck in it.
Okay, thank you.
We have one question that I caught in the chat. It was for Simon. The person asked me to read. So Jay-Z, who's a PhD student at Claudia's lab, asks, is forcing linked to another pathway like hippo pathway? And then could forcing rescue old adult fish?
Yeah, no, I answered that in the chat actually to her.
You did. Okay, so sorry.
We don't know about hippo. We're looking at that. We've made mutants in Yap and Taz, and we're studying them. But the lockdown is having the expected effect on those experiments. Yes. And no, we haven't looked at older fish. I mean, as I said in the talk, I don't really think that looking at aging fish is something I want to do. I don't think that an aging fish with its muscle wasting is really any better than looking at an aging mouse, and it's certainly significantly worse than looking at an aging monkey or person in some ways. So my view would be that the things we find in fish, we just go directly to the human population and look for people with variants in genes or differences in lifestyle that affect the pathways we're finding, and see whether that's implicated.
Yes, maybe we are part of a big experiment with this lockdown for a year now. We like our daily activities and caloric restriction and sedentarism. I'm asking questions. What are we going to see? Right?
And the absence
so we should definitely try to go.
Right? And the absence of exercise. I don't know about caloric restriction during this quarantine.
Right.
Like people are eating more.
Not
too
sure.
Much alcohol.
Yeah.
Yeah.
Too much
and
alcohol.
Sleep. Yeah, and also sleep habits. I mean, it's confusing, I would say. Anyways, I would like to thank all the three speakers for the wonderful talks this afternoon, everyone for participating and staying over time, and would like to invite everyone in 2 weeks' time. We will meet again to talk about animal models in biology and medicine in the Brazilian time, right? 6 p.m.? Sorry, Manuel, I forgot. I'm confused too. See? All right. I thank everyone, especially Professor Simon Hughes, Cláudia Mermelstein, and Veronica Salerno, and everyone else, and wish you a wonderful evening.
Thank you. Thank you. Bye-bye. Thank you for the invitation. Bye.
Bye-bye.
Thank you very much.
Bye. Thank you.